Dr. B.N.S. Murthy, respected Dr. B.N.S. Murthy, Director IHR, Dr. Muniappan, today's chief guest and speaker for this 100 months talk under AAPMHE, Association for Advancement of Pest Management in Horticultural Ecosystems. Respected Dr. Varghese sir, who is encouraging in this process and who is regularly attending all these online sessions, and all others who joined here. Some are yet to join. They are joining. In the meantime, we will go ahead with this inaugural session for this 100th months talk. To give a brief description about this one, we started this exactly 100 months back in August 2013 with the aim of cross-cutting learning among the plant production specialists. With that idea, we have started Plant Production Sciences Club at IHR. Since then, without interruption. That is the thing, because in our time stream, 25 years, 50 years, 75, 100, they have a special distinction. In that respect, we are in this gathering in the association, which is more than 25 years old, IHR, which is more than 50 years old, India celebrating 75th Independence Day. Now, in this function, though it is 100 months only, because of uninterrupted nature, we are taking it as a significant achievement. And in that respect, we are very much thankful to Professor Muniappan, who readily agreed for this distinct talk, who is a doyen in his own field. Before his speech, I will be introducing about the speaker. At that time, I will be talking about him. With this, I invite all the participants who joined in this 100 months talk, which is 132nd talk it will be. I invite all of you, and now I request Dr. M. Krishna Reddy, who is the head of the Division of Crop Protection at Indian Institute of Horticulture Research, to brief about the association and these talks. Thank you.
Good morning to all. Once again, welcome everybody for this 100th talk by Professor Muniappan, a well-known biocontrol expert and IPM expert for working for so many years. About AAPMHE, Association for Plant Protection in Horticulture Ecosystem, was started 20 years back with our founder president, Dr. P.P. Reddy, and founder editor again, Dr. Varghese. At that time it is started, and still now it is continuing successfully. And to this we introduce a program, monthly talks. Every month we organize a scientific talk on various aspects of crop protection. And these talks are delivered by students and even researchers, scientists of eminence, and whoever that period coincides, we organize, we collect information before and then put in a monthly talk. And this is continuously going on without interruption. Even during COVID period also, it is continuously organized. And about AAPMHE, see, the horticulture ecosystem, this is a unique society wherein we work exclusively for horticulture crops under different environmental situation, where how the pest scenario happening in horticulture crops. And it is see, other plant protection associations were specialized in dealing only specific situations, but this is being a unique and having different roles it playing. And the horticulture crops at present is, see, the change in climate and change in cropping pattern as leading to the several crop protection problems. So that is why we actually introduce the topics which are relevant to the current scenario, the pest scenario, and deal these things within a understanding of these situations, and disseminate the technologies which are readily available, at least for the farming community, which can help them to better manage the crop protection in horticultural crops. And with this, I thank the organizers and for this program and this support given by our director all the time. And the institute, we are successfully doing these programs, and we continue to take our director's guidance and further cooperation in this aspect. Thank you, one and all.
You have to unmute Sridhar.
Hello. Okay. Sorry. Thank you, sir. For the last 100 months, we have organized around 131 talks. We tried to compile all the talks along with the emails of all the speakers, and so that the audience can know who are expert in different talks, and if further consultation or some clarifications required, you can have it. With that purpose, we have compiled all the talks till now in the which were conducted last 100 months. Now I request our director, Dr. BNS Muthisar, to release the compilation of 100 months talks. Okay, thank you so much, sir. This compiled thing will be sharing to all the participants in due course. Now, I request to deliver the presidential address by Dr. BNS Murthy, sir.
Very good morning to all of you. With the pranams to Annadatas of our country and to the globe, I congratulate the society, the Association of Advancement of Pest Management in Horticultural Ecosystem, for such a wonderful and milestone issue on this particular day, the 100th month talk. Although it is 100th month talk, every month they used to have one specialized talk in the area of plant protection in horticulture system, and there are covered about 131 talks. This is a very heartening situation. And in fact, the challenges in the plant protection in horticulture is very, very important. When it comes to other crops, the major crops, there are quite a few workers also, researchers, scientists, and extension workers who can deliver the things. But the uniqueness of a horticulture crop, there are several crops, and each crop is really challenging. And its critical growth stages are also important. And many of these crops, especially some of the vegetable crops, where they need to go for harvesting in a very short period, maybe 3 days or 4 days, or sometimes alternate days, there it is very challenging when it comes to plant protection. When you are looking for residue-free production, if you are using any of the chemical pesticides, is very, very challenging. These things are taken very seriously, and the Society for Advancement of the Plant Protection here, they are coordinating this one. These talks, compile the talks, and that will be shared to you. And I am very happy that even without a miss of a single month, these talks are going on, so that to make it very light moment, that is called what is called Ishli. In Kannada, we call Ishta Bandaga. So this is not Ishli, it is regularly. That's what I see. And I congratulate this society for taking this things forward. And today's topic again, Muniappan is another great thing done in the biocontrol, biopest control and IPM practices. These days, there are several challenges in plant protection in terms of residues. What we see today for the clients or the consumers, they're asking for organic production. Organic production, that's why there are several chemicals which are used, which are not supposed to be used at a particular time. And the control of the pests and diseases is very challenging, as I told you. And because of this, there is a lot of residues, especially many of these cruciferous crops. and also some solanaceous crops, which are the major the basket of our consumption of fruits, vegetables. In fact, there it is really challenging, and there people are looking for organic production. Organic production, if you go for a pure organic production, it will be really not sustainable for our country with a population of 1.3 billion population. So there is need to go for a safe production of fruits and vegetables. In this scenario, biocontrol or IPM practices, what we are going to develop is very important. And with this this kind of a platform where you are exchanging the knowledge, views, and this particular directory that is getting compiled now, it will be shared with all of us, so that in future, if some expertise are needed in any of this area, they can be contacted and taken it forward. I will again congratulate this particular association for taking this such a nice initiatives in the field of horticulture, and this will go a long way in the protection of pests and diseases in horticulture crops. This area of control of pests and diseases is very, very important. If you see the records of the Kisan call center, where the questions are coming into for the services, pest and disease control is the major chunk of the questions that is farmers are asking, or even some experts and students are asking. This area is getting addressed. I am very happy with this one. And ICAR, ICAR-IIHR Bengaluru is always with this kind of initiatives. I look forward for many such important things and talks. I congratulate the society and also the speaker of the today's, and all of you for participating. Thank you very much for giving me this opportunity. Thank you once again.
Thank you so much, sir, for your presidential address emphasizing the importance of plant protection, whether it is horticulture or agriculture as a whole. Thank you so much. Now we'll welcome Madam Dr. Chandish Madam. I could see now many others are joining. Thank you. Now we'll go ahead with the presentation by Dr. Muniappan, followed by anybody would like to give their impressions will take it up after the talk. And before going to this today's talk on role of biological control and integrated pest management in India, for the benefit of the audience, I would like to give a brief introduction about Dr. Muniappan. Presently, he is the director of IPM Innovation Lab under CIRED, that is Center for International Research, Education and Development, located in Virginia state of USA. His basic expertise is on biological control of tropical weeds and insect pests, tropical economic entomology. He had his education BS in agriculture from University of Madras in 1963, MS in agricultural entomology from University of Madras in 1965, PhD in entomology from Oklahoma State University in 1969. He has a wide experience of traveling in, as a part of his job of extension and IPM. He traveled several countries and his expertise was shared in different countries and states like American Samoa, Micronesia, regions like Chuuk, Guam, Fani, Kosrae, India, Maldives, Marshalls, Palau, Papua New Guinea, Philippines, Rota, Saipan, Samoa, Tinian, Vanuatu, Yap, Nepal, etc. Coming to the brief summary of his experience, Dr. Muniappan is an entomologist who has specialized in biological control and integrated pest management research in the tropics for more than 35 years. His primary work experience has focused on the biological control of invasive weeds such as Chromalina odorata, Lantana chimera, Coccinia grandis, and Mimosa diplotricha. He also specializes in insect pests of tropical fruit and vegetable crops such as mealybugs, scale insects, whiteflies, caterpillar pests, and weevils. Dr. Muniappan worked as the Associate Director of Guam Agricultural Experimental Station for 20 years and also served as a consultant for the Food and Agricultural Organization. The Secretariat of the Pacific Community, GTZ, the Australian Center for International Agricultural Research and other international organizations. As the program director for IPM Innovation Lab, Dr. Muniappan is working with USAID, that is United States Agency for International Development, and project partner institutions in the United States and developing countries in Asia, Africa, Eastern Europe, Caribbean, and Latin America. In addition to his duties with Sridhar, he is currently serving as the chairman of the global working groups on Chromalina for the International Organization for Biological Control. As a major leader in his field, Dr. Muniappan has published more than 200 research and extension articles. This is in brief about Dr. Muniappan. There is a lot. With this, I request Dr. Muniappan to go ahead with 100 month talk under AAPMHE. Thank you, sir. Over to Dr. Muniappan, sir. Now you can share the screen and start presentation. Thank you, sir.
Is that okay? Well, thank you, Dr. Sridhar, for the invitation and such a long introduction. And also thanks for Dr. B.N.S. Murthy for inaugurating this seminar, and Dr. Krishna Reddy for giving us brief introduction on behalf of the Crop Protection Department, and also friends, and also happy to see some of my colleagues like Dr. Balal, Dr. Varghese, and other friends in this seminar. Since it is already, we have covered about 15 minutes in this session, I will go ahead and start with my presentation. Okay, the topic, as Dr. Sridhar mentioned, it is biological control in integrated pest management in India. So I will cover this topic in 4 sections. The first section, I will cover the history of biological control in India. It will almost form the basis for history of biological control in the world. This section is often not covered very well in the international scientific community presentations. Then the second one, I will be talking about biological control of weeds in India. There'll be a very, very brief presentation. Then the third one, biological control of insect pests. And finally, integration of biological control in the IPM program. To start with, biological control, yes, you all know, the term was coined by Dr. Harry Smith of University of California, Riverside in 1919. And he simply defined it as control or regulation of pest populations by natural enemies like predators, parasites, and pathogens. Since then, I think different definitions have come up with, but this is the simple definition that he came up with, and it still holds good. Well, as you all know, biological control has 3 major divisions: classical biological control, augmentative biological control, and conservation biological control. Classical biological control works well especially in managing the invasive species, because most of the invasive species get introduced without their natural enemies from the center of their origin. So by bringing in the natural enemies from the center of origin, we get fairly good results. One good example is the papaya mealybug control in India, and the other one is cassava mealybug control in Africa and also Southeast Asia. There are several examples, but I'm not going to go into the detail. Augmentative biological control, it has 2 subdivisions. One is the inoculative and the other one is inundative. Under inoculative, there is just one first release in this early in the season that takes care of, to manage the pests for the rest of the cropping season, like trichoderma treatment of these seeds before planting. And that Trichogramma inoculation builds up and takes care of the crop for the whole season. But in other cases like Trichogramma, we have to release them repeatedly once in 2 weeks or 3 weeks. That is inundative release. And this is practiced, I guess, for control of stem borers in rice and sugarcane in India. And there are several examples. Conservation biological control, that is preserving the existing natural enemies in this ecosystem. The major enemy to this conservation biological control is the use of synthetic chemical pesticides. Well, coming back to the history of biological control itself, I like to trace some activities from India. You know, the Spanish, when they colonized Central and South America in 1500s, they found the local native or the native population in these countries producing a dye. And that dye was produced by an insect that was feeding on the cactus. So eventually what the Spanish did, they started to produce this dye in Central and South America and exported that dye to Europe, and they made good amount of money or profit out of it. So the insect involved in producing that dye is the Dactylopius coccus, and the plant associated with that is Opuntia ficus indica. So these two are very closely related, and this insect produces very good dye. In the 1600s and early 1700s, British moved into the New World, and they saw that Spanish exporting this dye and making good profit. So they want to get into this bandwagon, In that process, they try to find out where the dye is produced, and the Spanish didn't want to give the information. They kept misguiding them, saying that they were collecting this dye from a plant. And anyway, then by snooping around, the British found that the dye was produced from the insect that is feeding on the cactus, but they didn't know the taxonomy of the cactus or the taxonomy of the insect. Well, they thought they can introduce this cactus into their colonies in the tropics like India, Sri Lanka, Australia, and South Africa. Then they can produce large quantities of this dye, and they can just defeat the Spanish production or overcome take over the market in Europe. So in that process, what they did, they introduced a cactus, Opuntia monacantha, to northern India. And also they introduced the Opuntia stricta and Opuntia elatea to southern India. And they tried to introduce different cochineal insects, but cochineal insects didn't produce very good quality dye. So they were not able to compete with the Spanish market. Yeah, but what happened, the cactus or cacti they introduced to India, they became invasive. So in northern India, all the way from Punjab to Assam, this Opuntia monacantha became a thicket and became invasive and a problem. Similarly, Opuntia stricta and Opuntia elatea became a problem in southern India, especially in the states of Andhra Pradesh, Karnataka, and Tamil Nadu. Then in the process of trying to find a good cochineal insect that will produce the dye, they introduced Opuntia monacantha into northern India. That insect didn't produce good dye, but it wiped out that invasive cactus in northern India. So that was the first incidence of an insect controlling a weed. Even though it was done serendipitously, that was the first incidence that happened in 1795. So coming to the timeline of the cactus biocontrol in India and also in the Indian subcontinent area, Opuntia monacantha became invasive in the 1750s. In 1795, introduction of the Dactylopius silonicus control that weed. Then they try to distribute Daclopia silonicus to southern India to control Opuntia stricta. In 1836, it didn't work because the silonicus didn't feed very well on the Opuntia stricta. Then the silonicus was introduced to Sri Lanka to control monacantha. It worked well. Based on that result, they introduced it to Australia in 1903. And eventually, they also introduced this Daclopius opuntiae from Sri Lanka to southern India, and that gave good control. So this is the picture. Actually, this picture, I took it over 2 years ago in Coimbatore area. There are very few patches you will see here and there. Still, the cochineal insect and the cactus do occur in very small patches. So Dr. Pruthi in 1969 stated that control of Opuntia monacantha by Daclopia silonicus in 1795 was not a deliberate attempt. Nevertheless, the potential of a biological control came to light for the first time using an insect to control a weed. Gaydon also mentioned the introduction of Daclopia silonicus to Sri Lanka in about 1865, and the successful control of Opuntia monacantha constituted the first international transfer of a natural enemy for biological control of weeds. However, Dr. McFadden in Australia wrote, Lantana was the first weed to be targeted for biological control and has been researched longer than any other weed. So similar statements exist in most of the European literature or even in the biological control literature. The work done in India has not been highlighted that much. And there is a need for the scientists in India to emphasize about this work, the early work done in India, and that is almost the first biological control of a weed happened in the world. That happened in 1795. Then another one that is misquoted in the literature is the introduction of Artesia insignis. Now they have changed the genus name as Insigno Artesia insignis. This insect is also neotropical in origin. I think the Kew Gardens in England was bringing in plants from different parts of the world. In that process, they have introduced this insect to Kew Gardens. And from Kew Gardens, they were distributing plants to other parts of the world. In that process, they introduced this insect to Paradinia Botanical Garden in Sri Lanka in 1893. From there, it got introduced to Nilgiris in 1915. But still, the scientists keep reporting that this insect was introduced to India for the purpose of controlling lantana. That is not correct information. That need to be correct information, citing from the previous information. So that need to stop. Another one misquoted is the introduction of Telnomus scrupulosa. It was introduced in 1941 for controlling lantana camera, but in the host-based testing done in the laboratory at Dehradun, they found it to feed on ticks, so it was recommended to destroy the colony. It was destroyed in 1943, but somehow it escaped the quarantine and established in the field. They found it in Dehradun area in 1951. Since then, it has been distributed all over India. And so far, it hasn't been found feeding on tick, but still you will find in the literature some of the scientists citing it as a wrong introduction because it is a pest of tick. Well, this gives the chronology of introduction of lantana natural enemies in India. It is quite sporadic, but I won't go into the details of it. But I have given my presentation to Dr. Sridhar. So if any of you interested in going through these slides, please feel free to get the slides from Dr. Sridhar. And coming to lantana work in the world, Australia was leading it. Then the second one was South Africa, and India has followed it. But the work has been sporadic, and it is a very sturdy weed. Even though Australia and South Africa have introduced over 2 or 3 dozen natural enemies, still they were not giving enough suppression of the weed. So right now, I don't think that much work is going on biological control of lantana in the world. Another weed that I want to mention in this presentation is the Cromolena odorata. I was involved working on this weed since 1983 onwards. Actually, I came to India to see what happened for the natural enemy that Parachaetus pseudoinsulata was introduced around 1975 to India. There it was reported as a failure. So I came to India to see what was the reason for the failure, because this weed has become a problem in Guam, where at that time I was working in Guam. So I came to see, and then eventually we were able to introduce Parachaetus into Guam and able to get good control. You can see it defoliated Cromolena thicket very well. And later on also we introduced this gall-forming insect, Cecidocaris connexa, into Guam. And also these 2 natural enemies were also introduced to India. This timeline shows the chronology of introduction of different natural enemies for control of Cromolena in India. So currently you have Cecidocaris connexa established in Bengaluru area, but still it hasn't been released in northeastern part of India, where Cromolena is still a serious problem. In fact, this Cromolena has moved from northeast part of India into Nepal, and it is a problem in Nepal too. And since I started to work on Cromolena around 1984 in Guam, we were successful in establishing Parachaetus, and that have good results. Then I started conducting international workshop. In fact, one of the international workshop in 1996 was conducted in Bangalore. And I continued that until 2006. Then I moved to Virginia Tech. There, Cromolena is not a concern, so I moved from Cromolena to Parthenium. Well, here is the distribution of Parthenium. As you all know, it is again a neotropical weed, got introduced to Australia first, and then India, then South Africa and Eastern Africa. We have a project controlling this weed in Eastern Africa. Well, you know about introduction of Trichogramma into India in 1983. Now it is occurring all over India, and it has also moved into Nepal and Pakistan. It is a fortuitous introduction to these 2 countries. Well, in Australia, they have been doing biological control of Parthenium for the last 25 years, and they have introduced, I think, 11 natural enemies. It is a hardy weed, and you need more natural enemies to tackle it. This slide shows the number of natural enemies introduced in different countries. Australia has introduced 11 of them. I think in India introduced only 1, but there is a fungus, I think the summer fungus, that also occurs in India. So there are 2 agents, and also there is a report of occurrence of the winter rust. So there's question about it, whether it is occurring in India or not. Some reports say no, some reports say yes. We have been working in Ethiopia for the last 15 years. We have introduced 2 natural enemies, and also the winter rust gotten in fortuitously. South Africa, they have introduced about 4 natural enemies. Well, I think I will give a pass for about 5 minutes, and if you have any questions, I will be happy to address it before I move to the next section. Is that okay, Dr. Sridhar?
Okay, sir. If only for this specific thing, if somebody wants to ask, you can ask. Remaining will take at the end, sir.
Yeah, remaining will take. If you have any, because there are a lot of, timelines and other information. If you have any specific question, I move on to the other biocontrol areas. If you don't have, we will continue with the,
Anybody?
I have one question.
Yeah, please.
I have one question.
please,
Can I go ahead?
please.
Yeah, I was
please.
just wondering, like, you use a biocontrol, some insect to control a weed, like the case of cactus like species in which an insect was used. So what would happen once, entire, all the plants are eaten up, then where would the insects go next?
Okay, I mentioned the introduction of insect in 1795 to India, especially to the northern part of India. The cactus is gone and the insect is gone. Or it may be there somewhere, but in very, very small quantities. You have to spend a month or two searching for it. That is the situation. They are specific.
Now, is there any risk like insect would change its host and may start feeding on economical plants?
I think some of our colleagues like Dr. Chandis Balal or others from the NBAIR could talk about it a little more eloquently, but there's, each country has its own rules and regulations, how to introduce a natural enemy, how to look for it. Some of the developed countries like Australia, US, Canada, South Africa, they have been doing this work for a long time. So to introduce any natural enemies, it almost costs about $1 million, because first of all, you have to find out where the weed or the insect that came in from, because mostly these are the invasive weeds introduced from one part of the world to the other part of the world without their natural enemies. So we have to find out the center of origin, then we have to establish a field, then identify the natural enemies over there, then we have to host specificity test in the center of origin. Once it is proven very specific, then we can bring into the country where we want to introduce. Then again, it has to go through the quarantine testing. So it is a very rigid procedure and it has lots of regulations involved in it. So fairly well controlled. Yeah.
Thank you.
Yeah.
Okay, sir. Please go ahead.
Okay. Thank you. Okay, I'm coming to the next one about the papaya mealybug. Papaya mealybug is also neotropical origin, especially it originated in Mexico. First, it was collected in 1955, but it wasn't identified as papaya mealybug or Paracoccus marginatus until 1992. Then around 1995, it started to spread from Mexico to the Caribbean countries and then to Florida, then to South American countries. At that time, in 2002, I was working in Guam. So it showed up in Guam in 2002. So I communicated with the scientists in the US and they mentioned about its invasion and availability of the parasites. So I introduced these 3 parasites. These parasites were picked up in Mexico by the USDA Agricultural Research Service scientists, and they hospitality tested, screened, and they were producing these parasites in Puerto Rico. So I imported that to Guam and introduced it in June. By September, it gave almost 95% control of the mealybug. Then around 2008, we had a program in India. So I came to Coimbatore to observe the program. And we found this mealybug occurring in the Tamil Nadu Agricultural University Botanical Garden. Then I went to Delhi and suggested to the administration that it is a new pest to India and they should take up biological control. But it took almost 2 years before they could come up or take up the biological control. By the time the mealybug has spread from Coimbatore to almost all over the southern part of India. And in 2010, they introduced the parasite, all the 3 parasites through the NBAIR. and they were able to establish that in the field by early 2011. They got very good control in this part of the world. The parasite not only controlled the mealybug in India, it also moved into other neighboring countries and suppressed the pest. For example, when this mealybug moved into Bangladesh or Nepal, they didn't even recognize it as a major pest because the parasite was suppressing it. And the collaboration involved, USAID mission in India, USDA APHIS, Indian Council of Agricultural Research, Director of Plant Protection, everybody in India involved in biological control, as well as some of the international organizations. Our economist, Dr. George Norton from Virginia Tech, was working on it, and he came up with a cost-benefit analysis of $500 million to $1.34 billion in benefits to India by introducing this parasite. Another mealybug that invaded India was the Solenopsis mealybug. Some people call it as cotton mealybug. There's the Phenacoccus solenopsis. It is a native of US. It was first described in 1895 from New Mexico State in US. It started to move around 1990. So it was picked up in Texas on cotton in 1990. Then it was picked up in South America and then Hawaii. In 2005, it became a problem in Pakistan, and 2006, it became a problem in India, and now it is spreading in Africa as well as in Asia. Then they found this parasite, Anaceus arizonensis. This parasite was picked up in India and it was described as Anaceus bombawale, and later on, the Turkish scientists found that bombawale is nothing but the arizonensis. So it was a fortuitous introduction to India, and it has taken care of the problem caused by this mealybug. Another insect, well, the previous two are on the classical biological control, this one is mostly the Solenopsis mealybug. We can't call it as classical, but it is a fortuitous classical biological control. In the case of Tuta absoluta, it is also known as the South American tomato leaf miner. Its center of origin is South America, but this pest got introduced from Chile to Spain in 2006, and then it moved across the Mediterranean Sea, moved into Africa, and then eventually it crossed the Arabian Sea, showed up in, I think, Maharashtra area. And then around 2016, it moved to Nepal and Bangladesh. I expected this to move from Nepal and Bangladesh to Southeast Asian countries, but this didn't happen. And also, when it is spreading from, say, Maharashtra to southern India, all of a sudden it showed up in Kathmandu as well as in northern part of the Bangladesh. The movement or the spread from one place to the other is not by the insect itself, as you see in the case of fallow mealworm. It is by the shipment of the infested tomato fruits. So I think some shipment from India, southern part of India, got into Kathmandu, and then from Kathmandu it started to spread all over Nepal. Similarly, it started in northern part of Bangladesh and then started to spread all over Bangladesh. Then it moved into Myanmar, and it didn't spread to, it moved into Myanmar, it didn't spread to Thailand and Malaysia so far. It has stopped right there since, I think, 2019, because there's not that much of shipment of tomatoes are happening from Myanmar to Thailand or Malaysia. But this insect has already moved in to the Central Asia, and then from there to China, and recently it was even picked up in Taiwan, but it is yet to spread into Thailand, Malaysia, and Indonesia areas. This shows the timeline of its spread. In the IPM Innovation Lab where I work, in Virginia Tech, we do conduct awareness workshops in the countries where we work. So we conducted some workshops in Kenya way ahead of the pest that moved into Kenya, in Nepal, Bangladesh, and others. These are some pictures of the workshop that we conducted. And so far we conducted several workshops just to create awareness as well as to inform how to manage this pest in different parts of the world. One of the insects, this is the Nishtocarus tenuis. It is myrid. It what we call a zoo phytophagous. This is a very good predator of the Tuta absoluta eggs as well as the small instar larva. And it is euphytophagous. If there are no insects, it will feed on the plant. If there are any insects like whiteflies or mealybugs or caterpillar pests, it will feed on them. Quite a bit of work has been done by Dr. Sridhar at the IIHR on Tuta absoluta, so I won't go into details of it. In our program, we did work and we have developed the IPM package or IPM management technology for Tuta absoluta, and we integrated it with the IPM package for tomato. Well, the next one I want to mention is about the fall armyworm, Spodoptera frugiferda. It is a neotropical pest. It got introduced to Nigeria in 2016. By 2018, it covered most of the sub-Saharan Africa. Then we expected it to move through the Middle East and to Afghanistan, Pakistan, then to India, but that didn't happen. It just jumped from Africa to Karnataka in 2018. From there, it has moved all the way to Japan by 2020 and also to Australia. But it hasn't moved in this direction that fast. I think only in 2019 or 2020, it moved to Pakistan. The southwest monsoonic winds seem to be carrying the insect fast in this direction. There is lots of work going on in Africa and Asia on managing this pest. There's some institutions like CIMMYT is working on developing resistant varieties to fall armyworm. But our program has been concentrating on using natural enemies or parasites and predators to suppress fall armyworm. And I guess even NBAIR is concentrating in that area. In Africa, even though it got introduced around 1916, the work on biological control was a little slow. But when it got introduced to India, Indian scientists quickly started to work on it, and they have identified so far over 15 or 20 different natural enemies. China has done even further more work. They have identified over 35 different natural enemies on the fall armyworm, and they are working very hard on managing this pest. Two of the egg parasites that we have been concentrating are, one is the Telnomus remus, and the other one, Trichogramma species. We found the Trichogramma vanjai that was originally described in Kenya is giving very good results. Gives about 60% parasitism. And also Trichogramma prettiosum seem to give good percentage of parasitism. And I think both of them known to occur in India, because sometime back, Trichogramma vanjai was introduced from Kenya to India, but that need to be checked whether it established or not. But prettiosum does occur in India. Our work in Nepal showed that Nepal currently has Trichogramma chylonis and also Telnomus remus. And we are trying to mass multiply these insects over there and trying to evaluate them in the field. There was some confusion on the origin of Telnomus remus. Telnomus remus was originally collected in Malaysia in 1935 and described it as Telnomus remus. Then Dr. Rao from CAB, Commonwealth Biological Control Station in Bangalore, went to Papua New Guinea and picked this insect and introduced to India. From India, he introduced to Israel, and then it was also introduced to Barbados and Trinidad stations. Then from there, it was introduced to all over the Caribbean islands, and then to South America, and then to Florida. In fact, around 2019, some of the scientists want to bring in this parasite from Florida to Africa. Now, we were not encouraging that activity because we know very well the parasite, when it was introduced to Israel, it might have already moved into Africa. In 1919, that information wasn't available, but later on the scientists surveyed all over Africa, and now they are finding this parasite well established in Africa. And also it has established well in all over the Indian subcontinent. So further work is going on in multiplication of Telnomus remus and releasing it for controlling fall armyworm. Here, this is a case of augmentative biological control. This timeline shows the introduction of Telnomus remus into different parts of the world. Another parasite we are working on is the Hebrobrocan hebitar. I think this occurs in India too. Sometimes it is called as Brocan hebitar. And also there was one publication from Bangalore showing that this parasite is working well on the fall armyworm, especially attacking the later instar larvae. And there is a publication from Hyderabad showing that Hebrobrocan brevicornis is also attacking fall armyworm. So these need to be clarified to see whether both the species are same or two different species are occurring in India, and their efficacy on fall armyworm need to be evaluated. And the best one need to be introduced, mass multiplied and introduced in the field for suppressing the fall armyworm population. We have been working in Niger for controlling one of the pearl millet pests. We were using the Hebrobrocan hebitar. There actually the simple technique was designed by one of the scientists, Gaukar. He was working in 1980s in that region, and he came up with using a gunny bag sack. In that, he produced some broken grains, either sorghum, pearl millet, or maize, and released some Coursera cephalanica larvae inside this bag. In addition, he released 2 pairs of the Hebrobrocan parasites. Then they tie this bag in the field. This picture was taken in Niger under their condition. They also engaged the farmers to take these bags and place them in their field. Here they were placing these bags under a small bucket so that the bags won't get wet when there is rain. Sometimes they use even their cut gourd as a protective measure for these bags. And they tie them up in the field in different areas. Here they are tying up this bag on the top of a little hut. So another method we are trying is trying to produce these parasites in a nuclear center that is maybe in this slide was prepared for Nepal. So we try to set up a nucleus parasite producing center in the capital area like Kathmandu. Then we try to develop satellite centers in different provinces or states. And these centers could be university centers or some centers or the cooperatives like rice mill cooperatives or maize mill cooperatives, private entrepreneurs, anybody who is willing to produce the parasites and provide to the farmers, we try to help them. But this nucleus center serves as the backup. If there is any problem in these satellite centers, this nucleus center will get involved and help them out. And also nucleus center provides training for these satellite centers. So this setup or system we are trying to introduce to different countries. Of course, India is such a big country, so probably this need to be considered on a statewide basis. And also in the augmentative biological control, when we were introducing for the fall armyworm, there was quite a bit of resistance to it from the scientists, especially those who are promoting synthetic pesticides. In the analysis I came up with, we can even call the use of chemical pesticides as augmentative control, because we have to repeatedly spray the insecticide in the field. So that means we are augmentative. So it is augmentative. So that is, recently we published a small blog in the Entomological Society of America on this aspect. And we are getting some response from different scientists giving different views. Also, I will stop over here another few minutes if you have any questions before I go into the integrated pest management and integrating biological control into IPM. Sridhar?
Okay, sir. If anybody wants to ask for any clarifications, please go ahead. Anybody have any questions?
Sridhar, I have a small.
Yes, sir. Dr. Varghese, sir, please.
Please.
As I was discussing with you, Dr. Muniappan, even before we started the program, for example, take an example like this Solenopsis, which is very effective,
Yeah.
And I tell even we have published a paper on this along with Chandish Balal in Current Science in 2016.
Yeah.
Excellent. Now, the issue that we often face from the extension agents as well as from the farmers is the fact that they ask for, I'm not going to call it inundative or whatever, I'll just say interventional releases of Solenopsis at the appropriate time in a specified period in a specified area, and which is in volumes, and we do not have that. We do not have that capability in terms of commercial supply. So do we have to beat these drums often and say, yes, we are doing this and everything is working fine? And I know the farmers in India, I'm talking from a real scenario, during the fruiting season, and when the pinhole boring starts, which is the later phase of infestation of tuta, they invariably go for an insecticide, say, like flubendamide, say. And once that is sprayed, the entire gamut of all the bouquet of all the parasitides, everything disappear. And then these two don't get along. But in insecticide, for a serious tomato grower, we have more than 800,000 hectares of tomato in India. Insecticide continues to be the mainstay because we do not have suppliers. Suppliers like Copper or BioB world over, they all supply. We do not have that. And one of the area, my suggestion is, we have mass rearing technology, which doesn't fit the bill. We need to have commercial upscaling technologies are needed, like what the Copper does. Only then, these issues become relevant at the grassroots ground level. That's my remarks. Less of question, more of remark. So maybe you can respond to that. Thank you.
Dr. Varghese, well said. That is true. But, this augmentative biological control or release of this parasite work is going on well in the developed countries, especially under greenhouse conditions in Canada and to Europe. Several of them adopt this. In fact, they program, and say in cucumber crop, when to release these parasites or the predators, things like that, that has been programmed. And then in the field condition, it is somewhat difficult, but still in the European condition, like Trichogramma evanescens has been used for controlling Tuta absoluta. And that is available on commercial scale, and it is going on. But in developing countries, it is, the problem is not all the farmers are that well informed. The extension system also is somewhat limited. And then the production happens under the government, mostly government laboratories that need to be taken into the private conditions as well as into the extension area. If you look into the Trichoderma distribution, I've been involved a little bit at the Tamil Nadu Agricultural University because we had a program over there from 2005 to 2014. So there you can see the trichoderma being produced by the Tamil Nadu Agricultural University and selling it to the farmers. Then the KVKs are producing and selling it. And also private entrepreneurs also producing and selling it. So the fungal agent could be kept for 3 to 4 months or 6 months in the storage. But in the case of these parasites, we have to find the demand when we have the supply. If you don't match the demand and supply, it will be a problem. So these things we have to.
That's an area of research, that's a researchable issue. See, Trichogramma and Trichoderma are not comparable, but Trichogramma and the Solenopsis or any of the macrobials, the parasitoids are effective provided we have a way out, I think that has not been modeled into, I think we need your help and suggestions on that and stimulate the private people to get into it,
Last week I was in Nepal because we have a project in Nepal. We don't work in India in this current phase. So there I was trying to implement that model that I showed you in mine. And we are producing the parasite in Kathmandu and also Rampur area where the government labs are there. And we are giving training to the provincial laboratory people. Still, it will be a long way to go. Then I went to one of the rice mill owner to see whether he will accept the idea of releasing these parasites in the rice because he has about 4,000 farmers who supply to his rice mill. He was very smart. He said, yes, I can buy the cards and give it to my farmers, but I will buy only during July to September, because that is the growing season for me. That's when I need it. The rest of the time I don't need it, so I can't be producing it. So you have to figure out a method how I can get the cards at that time. So we have to integrate lots of these things. We worked on releasing that Hebrobrocan hebitar in Niger. Niger, they grow only pearl millet. Pearl millet is grown only from June to September. Rest of the time, it is very dry. Nothing is grown in Niger area. But there is one caterpillar pest that attacks the earhead that stays in the soil until August and comes out from the soil and lays eggs on the pearl millet earheads. And the caterpillar feeds on the earhead. Then it falls to the ground in September. Then it will remain in the soil. So we have to come up with a method how to bring in the parasites during that particular season. So we worked out with a farmers co-op to produce. We will give the inoculative culture in, say, May to the farmers co-op, and they will start producing and releasing it in August. And then, of course, after September, they won't be doing anything. Only we have to keep the culture in the laboratory until next May. Well, so it varies from place to place and depending upon the pest and the crop.
Yes, you're very correct. I was talking always in the context of India, and I would also like to know what is your take on the pesticide industry? They are equally powerful in their extension.
Okay, in my work, the pesticide industry is not very happy with our recommendation. So I also counter them, recently. Some of the scientists were saying that augmentative biological control is very expensive, not cost effective. Then I found that in whatever parasites or predators produced in a country, you are developing the capacity of the human capacity by training your scientists and technicians, and also developing your laboratory capacity. So we are developing the human and institutional capacity in the developing countries by going through this augmentative biological control process. In the case of chemical control or synthetic chemical pesticide use, most of the pesticides are produced by developed countries. So over 80% of the money spent in the developing countries on chemical pesticides go back to the developed countries. Only about 20% or 10% of the money spent on pesticides stay in the country. That is just by the distributors. So that is the current situation. Not very many pick up that scenario.
Thank you, sir. Remaining discussion, we'll have it after
At
the
the end,
presentation at the end. Thank you.
Okay, thanks.
Thank you.
Okay,
Thank you
this
very much.
slide shows the countries where we work in the current phase. These are 7 countries the IPM Innovation Lab is working in currently. 3 countries in Africa and 4 countries in Asia. And we have 8 projects. I won't go into the details of it. And we do concentrate on the IPM packages and involving biological control in those packages. We work on vegetable, cereal, legume, and fruit crops. The way that we address is we develop IPM packages for the selected crops. We also monitor and manage invasive species. Then we provide graduate training as well as short-term training to the farmers. That is the mandate of our project. What I mean by IPM package, in the past, our scientists used to work, one will work on a pest of tomato, another one will work on on a disease of cabbage, and another one will work on an economic threshold of maize or something. But we asked our scientists to come up with a package for each crop that we are working. What we mean by that is we wanted our scientists to address the problems faced by the farmers from the time of planting the seed up to the harvest by coming up with alternate technologies to solve those alternate technologies instead of using pesticides to solve the problems. So this is a list of components for the IPM package for tomato. It is only a template. It may not apply in all countries, in all situations, but the scientists can pick and choose whatever they think that is appropriate under those conditions, like tracing healthy seedlings, treating the seeds with Trichoderma, Pseudomonas fluorescens and all, because they are beneficial ones. So these are the different components we recommend for the tomato IPM package. Similarly, we developed packages for other crops that we are working. Here is a slide showing Trichoderma production. This was taken in Bengaluru, and in Bangladesh, they make Trichoderma compost in the backyard and they use them in the fields. We also introduced this eggplant grafting to overcome the bacterial wilt disease of tomatoes and eggplants. And this was introduced into Bangladesh, and then we were also working with Tamil Nadu Agricultural University, but it is more of a problem in the humid tropical areas like Kerala and into the Bangladesh area. Of course, we all know we also recommend traps, pheromone traps, and this used to be not that popular about 10 or 15 years ago in the developing countries. Now it is very popular. This slide was also taken in India from BCI production on the NPV. We are also looking for NPV for fall armyworm. I think in India you have already found it. In Bangladesh, they are importing a commercial formulation of NPV for fall armyworm. It is known as Phalagen, produced by an Australian company in US, and that has been registered in Bangladesh. But in India, I think you should work on finding the local NPV for fall armyworm and use it for controlling it. This slide was taken some years back in Indonesia. There they were producing Podoptera litra NPV in the laboratory and supplying it to the farmers. We also recommend production of egg parasites, larval parasites, and here it is a simple technique of collecting the predaceous mites. This predaceous mite production and export is a very big business in Kenya and Tanzania. The European companies there produce quite a large amount of these predaceous mites and ship them to Europe because there is a good demand for the predaceous mite for controlling phytophagous mites on the ornamental plants. We also recommend use of neem. I don't need to go into details of it because lots of work has been done in India. Some work is being done on a very crude form in Indonesia and producing the neem extracts. Another thing that we address is the virus disease, peanut bud necrosis virus. It is a serious problem in southern India. This picture was taken in Coimbatore, and we recommend roguing of the seedlings in the nursery itself, so that the infected seedlings won't go to the field. If there are no infected seedlings, the thrips, it is transmitted by thrips. So thrips won't have the inoculum to transmit from one plant to the other. So if we rogue and plant the healthy seedlings in the field, then we get a very good crop. If not, this is what happens in the field. And also we recommend what we call host-free period for controlling the tomato yellow leaf curl virus. This technique is used mostly in Africa and in the Caribbean islands. The tomato leaf curl virus is transmitted by Bemisia tabaci. If you don't grow tomato or pepper in an area for about 3 months, even though Bemisia has several host plants, it won't have the virus inoculum to ingest and transmit. Then when we plant after 3 months, the crop usually grows well and without the disease. And even though the disease comes in the later stage, it won't cause that serious a problem. But it all depends upon how to implement it. It is easy to implement in Africa because there the crop is grown in small patches, is surrounded by desert. So there's not much of the population of the Bemisia coming from outside, but it may be very difficult to implement in India. So this slide primarily shows how we develop the management technology for, first we developed the IPM package for tomato. And when Tuta got introduced, it disrupted the whole package. Then we have to come up with the management technology for Tuta, then integrate it with the IPM technology for tomato. So that's what we did for the tomato. Similarly, that is what need to be done for fall armyworm. Before fall armyworm got introduced to Asia or Africa, there's very little problem, pest problem in maize crop. But since its introduction, it has become a major threat. So now we have to develop a management program for fall armyworm and integrate it with the management of the IPM package of the maize. So in general, there is confusion. Some scientists say, I am an IPM specialist for thrips. That can't be the case. IPM is for a crop or a cropping system. It is not for a particular pest or particular disease. There we have to develop a management program for a pest or a disease. Then we have to integrate it with the cropping system or the crop management, IPM for the crop. So that's all. I want to thank Dr. Sridhar for inviting me and Dr. Asokan for facilitating, and also thank you all for participating in this seminar. And if you have any questions, comments, please feel free to.
Thank you. Thank you so much, sir, for your excellent presentation, particularly focusing on the history of biocontrol and what should be the IPM and various aspects of biocontrol based on your vast experience. Thank you so much. I was watching your local time clock. You started on 8th as per your local time. Now you have entered into the 9th. It is already midnight for you. Thank you so much for having patience during midnight, and now we'll be having some interaction. Whoever is having small focused questions, kindly ask for clarifications. Kindly let it be a focused question. Thank you. Dr. MS Yadav.
Yes, sir.
Yeah, please go ahead with your question, please.
Sir, my question was because this Parthenium hystrophorus is a very serious problem in India, and sir was telling about a rust or a fungus for control of this weed. Because Zygogramma bicolorata has introduced in India, but which fungus control this Parthenium?
Well,
Hmm.
shall I answer?
Yeah, please.
There are two fungi. One is the winter rest and another one is summer rest.
Rest.
I think the summer rest was already fortuitously introduced to India. It came by accident. It wasn't introduced by the scientists. It is there. And very recently, the winter rest was reported from Nepal. I think it is also in India. I was looking into, about a couple of months back, we had a webinar on Parthenium. In that, Dr. Bhaktavachalam from NBAAR participated, and he gave a list of different natural enemies introduced for Parthenium in India. And also Dr. Dileepan from Australia gave a presentation. So in Australia, they have introduced 11 natural enemies. I think Dr. Chandis Balal was trying to introduce one or two natural enemies from South Africa. So there is need for concentrated effort to bring in some of the natural enemies that are already used in Australia to India. That has to come through your NBAAR.
Okay, thank you, sir. The next, Dr. Chandish, madam.
Thank you, Sridhar. Thank you, sir. Each time it's a great learning experience from you, sir.
Well, thank you.
I want to just share a couple of thoughts and a couple of questions quickly, sir. One is regarding the point which Dr. Verghese raised also on the availability of macrobials and microbials. And the technology is there with the organizations. The microbials and macrobials are also available with the production technologies. Now, even in Africa, I'm sure this kind of problems are there with respect to availability. In case of microbials, it is the question of developing the toxicology data, which is very expensive. In the case of macrobials, it is the problem in the companies ready to accept the technology and to mass produce it, upscale it, for example. So a couple of technologies which I developed, in fact, I have just prior to retirement, I told the then director that it can be removed from the commercialization list and included in the training module so that more entrepreneurs can at least pick up the technology. So in your opinion, how can we really take up these production technologies? Because it's not the responsibility of the research institute to upscale and supply to all the farmers. It is impossible, though for demonstration trials, we are doing it. So in your opinion, how can a company take it up? For example, UPL had taken up the technology of Trichogramma production. So similarly, can even the chemical pesticide companies be motivated to take up the production of microbials? Because we used to keep telling them, when you have the facility and expertise, why don't you take up at least a couple of biopesticides so that you can ensure that it is available? So is it feasible for bigger companies to take up production of microbials primarily, if possible macrobials, so that it becomes available to farmers?
Because of the publicity about the synthetic chemical pesticides causing adverse impact in the environment and other problems, many of these companies are also looking into developing the microbial pesticides. Of course, long back they developed the Bacillus thuringiensis one, and it is widespread all over the world and it has well established. And recently the Phalogen, I think Agrobiotech or something in Texas company, but originally it is from Australia, is producing Phalogen. And they registered it in Bangladesh and also in Sudan. And they are very active promoting it. But the problem is the cost. how many of the poor farmers would be able to buy that and use it? So if the NPV for fall armyworm is produced locally, just like what happened in the case of NPV for Spodoptera litera, around 1990 or so, I saw in Rahuri at the Maharashtra Agricultural University, they had a small room wherein they were producing large quantities of NPV for Spodoptera litera and distributing it to the farmers. It was a crude form, but it was cheap and it was working. I think right now what I'm hearing from like the PCA and others who used to produce NPV for Helicoverpa and Spodoptera, they say there is no market for them, so they are not producing. So even the big companies are looking at producing these microbials, and of course they will get involved only if they can make money out of them. But in the developing countries, it is a different scenario. We have to even in the US, registering Trichoderma is very expensive. So what they are doing is they are putting it into the compost and selling it to the public. There is no registration required. So we have to come up with alternate methods how to skin a cat by different method. And also we need to include the policymakers in these discussions and also the regulatory agencies. So if we work, the researchers working with the regulatory agencies and the policymakers, that's where we can make a big progress. We need also to involve some of the progressive farmers so that they would be the advocates for us. For example, when I first found, that papaya mealybug in Coimbatore, actually I went to ICAR and talked to some of the administrators. One administrator said, well, it is due to climate change. So he showed the door and asked me, never mind about the papaya mealybug. And another administrator, when I talked, and he said, mealybugs are known to occur in India for hundreds of years. This can't be new. Then what happened from Coimbatore when it came to Karnataka started to affect mulberry. There they can't use pesticides. When those farmers made a big hue and cry, then the government started to listen. So we have to, use all these different resources to convey the message and carry out our program.
So just a couple of points too. For Trichogramma long-term storage, we have a technology which we have published in Canadian Entomologist. You said the availability during a particular season. This technology enables 3 months of storage, sir.
Oh, that's
and
good.
Point number 2, NPV local isolate for fall armyworm is already available with NBAIR. And Trichogramma pretiosum is not indigenously found in nature. It is not established on fall armyworm eggs, whereas Trichogramma chilonis and Telenomus remus both are established in the field. So these are, and one more point I wanted to say, though Esophagus papayae established against papaya mealybug, following a recommendation of total abstinence from chemical pesticides, a similar situation could not be seen for cotton mealybug, sir. Because Inaceus bambavale, though we do record parasitism, it did not establish, is it related to the heavy pesticide load?
That's
These
good.
are a couple of points which I,
Yeah.
thank you, sir. Thank you so much.
I just want to mention an incident when this cotton mealybug, Phenacoccus solenopsis, got into Pakistan and India. At that time, some scientists contacted me whether I can facilitate getting some parasites from California. So I contacted Gillian Watson. She used to work at the Department of Agriculture over there, and they said they will give the parasite. At that time, the mealybug was a problem in Israel, Pakistan, and India. Then they said, we will give this parasite culture to only one country, and that country has to mass multiply and provide to the other countries. I said, this is the best situation you can come across wherein none of these 3 countries will cooperate with each other. So meanwhile, that didn't happen, but the parasite itself came in fortuitously. Well, that's a side story. Okay, any other questions? Please?
Any other questions, please? In Thank
Yeah. Only again, I want to make one remarks, that is taking forward the Chandish Ballal's the concern. In fact, our system of cultivation of different crops, again, the question of small farmers come into picture. And in Western countries, large scale of individual few farmers are taking up. If we can convince them, and the seasons also very limited there. So the mass production is possible and store. Our technologies also should be such that whatever the parasites that we develop, we should be able to multiply in a very short time. That is where our success stays. And also the threshold level of this particular pests, it has to be looked into and released at a particular time. Our extension methodology should gear up, and also again the policy of introducing some of these things is to be concerned. Then we will be success in many of the biological control systems. That's what I wanted to make it.
Well, good point. Coming to the release of the parasites, for example, I monitor the publication or the annual reports of the national IPM. They are releasing Trichogramma japonicum for control of the stem borer of rice. So that activity must be there. So I'm not sure how popular it is, but I see in their annual report saying that it is giving very good results. And recently I got a note from one of the scientists from Texas, A&M University. He says releasing the egg parasites alone not necessarily will suppress the population of the pest, because over 80% of the eggs of these moths always get damaged by the abiotic and biotic factors. The 20% or 10% of the caterpillars that escape, they become serious problem. So releasing the egg parasite is not contributing that much in controlling the pest. His study was done in cotton system in Texas. But of course, that is his opinion and his research. But here, what we are recommending is releasing Telenomus, Trichogramma, and also Hebrobrocan, because Hebrobrocan will attack the later instar caterpillars. In Kenya, they have a Cotesia ECP that attacks the early instar caterpillars. So there they can release Trichogramma, Telenomus, Cotesia ECP, as well as Hebrobrocan to tackle the fall armyworm problem. Well, there's lots of this integration between the government research, government extension agencies, private industries, cooperatives. All these need to be integrated, and we have to find out a method, and that is, that is profitable. Anybody will get involved only if they find it is beneficial. Farmers will use the parasites and predators if they think that is giving good control, and also they can, profit out of that.
Okay, thank you, sir. Anybody else?
Sridhar, I have one more suggestion.
Please.
Please, sir.
I think we have not got everything which is the real potential of Dr. Muniappan, So I think you should reserve him for a future, because there are many things like fruit sucking moth in which he's an expert, and we need, some. So I'm giving a suggestion that you need to, slot him for a future time, and Dr. Muniappan kindly, come to us again.
Thank you for your session, sir. Now it is open for Dr. Muniappan. Whenever he is free, we are ready to facilitate and for sharing your knowledge. The platform is mainly meant for sharing of the knowledge. We are very much thankful for coming forward, sir. Not only you, whoever has joined here, successfully you could do 100 months. Why can't we continue? Let us not keep any specific target. It should continue with the good motive. I request all of you to come forward to share your knowledge, just send a mail. We'll facilitate. Now it is open, unlike earlier. We are reaching to different countries. Today, participants from Bangladesh, Nepal are there. Several ICR institutes have participated, which was not possible earlier. Let us share and let the plant production knowledge be shared with by updating whatever is happening. Sometimes we may not be having a small idea, which is very much useful outside. Suppose today morning we have to search one farmer. Morning 7 o'clock he called he's from Guntur. He's having a specific problem of thrips on chili. When I suggested spinosad, he told whatever I suggested, double dose also I used. Dr. Madhu Reddy madam has suggested my number. Then I thought I will consult Dr. Rachana. She is in this meeting. After this, I'm going to send her the photo. He says it is not looking like a thrips, but it is thrips only, looking very big. And if it is a thrip, whether we have to revise the dose or still better molecules you have to suggest, because you cannot start doing research and suggest them. He says that particular farmer, if this is the case, our crops cannot continue, particularly chili, he told. By today evening, we want to give him some solution. That sort of small discussions, I may not be knowing, some of you may be knowing for this particular problem. Ultimately, we should be able to give the solution to the farmer. With that motive, I request all of you to suggest your friends also, whomever throughout the world, if you are knowing somebody in a plant production field, any field of either entomology, pathology, nematology, any field, you can suggest and take their appointment, and let us have this type of sessions for the sharing purpose. Thank you. Now, anybody else is there? Otherwise, I profusely thank Dr. Muniappan sir for your excellent presentation and particularly sparing your midnight time there. Now, as a formal vote of thanks, I request Dr. Uma Maheshwari to give. Next month, we will be joining with 101st month pack. Before that, I request Dr. Uma Maheshwari, who is the secretary of our association, to give a formal vote of thanks. Thank you.
Good morning, everybody. And it's my privilege to have been asked to propose a vote of thanks on this historic occasion of 100th month talk of AAPMHE. I, on behalf of Association for Advancement of Pest Management, Horticultural Ecosystems, and the entire IIHR fraternity, I express my deep sense of gratitude to our guest speaker, Dr. Muniappan sir from IPM Innovation Lab, Virginia Tech, USA, for gracing this occasion and also delivering the special talk on role of biological control in integrated pest management in India. Thank you, sir, for enlightening us with your vast experience and enriching our knowledge with new insights on biological control and IPM. And I sincerely thank Dr. B.N. Srinivasamurthy, sir, our director, ICAR-IIHR, for his endless support and constant encouragement for the conduct of this program. And our heartfelt thanks to Dr. M. Krishna Reddy, sir, Head Division of Crop Protection, ICAR-IIHR, for his adept guidance. And I extremely thank Dr. V. Sridhar, sir, President AAPMHE, for shouldering this responsibility and taking AAPMHE to greater heights by successfully organizing the scientific talks for 100 months without any break. And we greatly acknowledge the valuable participation of our founder AAPMHE members and seniors, Dr. Abraham Verghese sir and Dr. Chandish Vallal madam for their valuable inputs and also in streamlining our AAPMHE activities. I take this occasion to thank all the speakers who delivered scientific lectures in our plant protection club so far, right from the beginning first month to 100th month today, and share their knowledge and experience on various aspects of plant protection. And I extend my sincere thanks to all the participants of the meeting from IIHR and other ICR institutes, Dr. Shahadat sir from Bangladesh and Nepal, and ICR institutes participants from CIWT Bhubaneswar, IRA New Delhi, CPCRA Kasargod, NABSM Raipur, then DGR Pune, NCIPM Delhi, IASR Lucknow, NBAR, and KVK Dharwad, and all other participants from ISCIUs and institutes for their active participation through the Zoom. And also this obviously shows their interest in this topic. And we acknowledge thankfully the efforts of our staff from KKMU for their assistance in arranging this online program successfully, and my sincere gratitude to all those who are directly and indirectly involved in this event. And thanks once more to one and all, and thank you very much.
Yeah, thank you so much. Thank you, everybody. We'll be closing this meeting and we'll be joining in the next month. In the end, a special thanks to Dr. Muniappan, sir. You
Thank you
started
very
your
much.
talk on 8th of November. You reached 9th of November. Now just you reached 9th of November. It is already very late for you. Sorry for that. But all of us got benefited. With this, I thank from our association side everybody, and we are closing this meeting. Thank you so much.
Thank you.
Thank you.
Bye.
Thank you.